Hi. Good morning, everyone. Thank you for joining me today. I'm excited to share insights on a topic that is rapidly transforming the drug discovery landscape, which is targeted protein degradation, or also known as TPD. So TPD has emerged as a game-changing approach, going beyond inhibition to eliminate disease-causing proteins. So it unlocks previously undruggable targets and promises to transform therapeutic strategies. But success in TPD requires more than just innovation. It demands rational drug design and systematic execution. So that means that understanding of the biology and chemistry and applying robust screening methodology, predictive modeling, and iterative optimization to enhance efficacy and specificity of the degraders. So I'll go into the details now. So today, we will explore how rational drug design and systematic execution accelerate degrader discovery. So I will highlight key strategies employed in our in-house case studies here. So these are real case studies that show how scientific methods and strategic planning come together in practice. So I will walk you through two real-world case studies, one on a V1 molecular glue and another one on a PIM3 bifunctional degrader or PROTEC. So these examples are more than just a compilation of assay data. They are examples telling broader stories. So they showcase our thinking process and also our ability to successfully perform these assays in-house. So they are a window into how we apply the tools and capabilities in the box here to solve complex challenges in targeted protein degradation. So from high throughput physics, biophysical screening, and AI-driven design to robotics-enabled chemical synthesis and translational cellular assays, you will see how each capabilities play a role in accelerating the discovery and improving the decision-making. So the goal here is to show you that not just what we offer, but how it all comes together to help you to move faster, reduce risk, and ultimately get better candidates into the clinic. Yep. So let's take a look at the capabilities that really make a difference when working on targeted protein degradation programs. So what you see here is just not a list of services. It's a toolbox that we have built up to help scientists to tackle unique challenges for TPD. So on the left, whether it's designing or synthesizing bifunctional molecules, identifying binders for E3 ligases, or characterizing ternary complexes, we have developed solutions that are both scientifically rigorous and practical to implement. So for example, our comprehensive chemistry solutions enable us to synthesize and optimize every component of the PROTECs, from the linker design to warhead and the E3 binding, E3 ligand binding site. So entirely, we can do this in-house. And our biophysical platforms like spectral shift and surface plasmon resonance, or SPR, help us to understand complex formation in real time, complex molecular interaction in real time. And then for cell permeability and ADME properties, which can make or break a degrader. If you can't get in, then you can't do anything. So we know that this is important, so we have integrated it early in ADME profiling to help our clients make smarter decisions faster. So our cell-based assays like splinter protein turnover assay and in-cell target engagement assay will give us insight into what is happening inside the cell. So starting from the next slide, as we go through the case studies, I will show you how these capabilities come together to support discovery, not just in theory, but in real-world projects. So this is our first case study. This will focus on molecular glues. So you know that these are small molecules that stabilize protein-protein interactions. So screening for molecular glues for a protein target is challenging. So we introduced a new high throughput spectral shift assay to rapidly detect binary and ternary binding events simultaneously. So V1 here is a key oncology target So using this approach, we identify V1 degraders and demonstrating how scalable screening work can accelerate glue discovery. So now let's take a closer look at V1. So this is V1 is a critical regulator in the cell cycle. V1 is a nuclear kinase that plays an important role at the G2M checkpoint. So its job is to make sure cells don't just rush into mitosis before they are ready. So specifically, it inhibits CDK1, which helps delay mitotic entry and give the cell time to repair any DNA damages. So this is important because entering mitosis with damaged DNA can lead to genomic instability or cell death. So V1 is acting like a gatekeeper, ensuring the cell only proceeds when it's safe. So from a therapeutics perspective, V1 is a very important target. So inhibitor like adavosertib here, also known as AZD1775, has been shown promising result in both preclinical and clinical settings, but unfortunately, it's not without challenges. It has a very narrow therapeutic window, and the toxicity can be a concern. So probably due to its not so selective kinase selectivity issue here. So PIM1 is here. It also targets other kinases here, so that's why it has toxicity issue. So for this reason, we think that TPD approach, the protein degrader approach that doesn't target the ATP binding pocket site would be advantageous. Yep. So before we go into the case study, let me give you a quick comparison between molecular glues and PROTAC. So if you are familiar with PROTACs, you'll know that they are designed with an intent. They are medium-sized molecules that binds to both the protein of interest and also the E3 ligases. We can measure those binary interaction here, on either side, and then we can predict something called cooperativity factor and also predict hook effect at high concentration. So this is a very rational and structured approach. But molecular glues, on the other hand, is a totally different story. They are small, drug-like molecules, but they often don't show strong binary binding. They don't show binding to usually the protein of interest. So instead, they work by stabilizing a ternary complex. So sometimes with cooperativity factors of greater than 50. So what is cooperativity factors? Cooperativity factors tell us how efficient the two proteins interact in the presence of the glue compound. So the larger the value here, is better. It's better because it means that we get smaller KD for the ternary complex, and which means better binding for the ternary complex. So essentially, it's a measurement of how well the three components come together. So cooperativity factor of 1 or greater than 1 indicates that the presence of one binding partner enhances the binding of the other, which is ideal for degraders. And unlike PROTACs, molecular glue doesn't exhibit the growth hook effect. The challenge here, however, is traditional high-throughput screening methods are not built to detect this kind of mechanism of action with molecular glues. Yeah. So we need a new tool or new tools. So ones that can pick up on functional outcome like induced ternary complex formation or induced protein-protein interaction, even when the binary binding or the binding to the protein of interest is weak or even absent. So this is where innovation in assay design becomes critical. So this slide shows our strategy for identifying molecular glue. So we have designed 3 parallel screening conditions to cover all the key interaction possibilities. So in condition 1, we focus on identifying site-agnostic binder to V1. Here, we measure the binary dissociation constant or KD to access direct binding to the protein of interest V1 here. Okay, condition 2 is the same as condition 1, except that we are measuring the binding to cerebron or CRBN. So again, using KD value, measurement of KD to confirm that there is binding to the CRBN and the measurement should be site agnostic. So condition 3 is where things get interesting. We look at ternary complex formation between V1 and cerebron. So this allows us to measure both the KD of the ternary complex and the cooperativity factor. So cooperativity factors again tell us how efficient the two proteins interact in the presence of the glue compound. So this setup here, these three parallel screening conditions here are intentionally designed to be simplified. So we use only one label protein, yes, which is we label only V1, which help us to reduce false positive compared to traditional biochemical assays. So running all three conditions in parallel maximizes our chance of identifying genuine binders. I will show you how later. So it also allows us to discriminate binding efficiency for each of the partner. And thanks to something called cooperativity factor, we can calculate each of these individually. And the best part is the entire screening here can be completed in just one day or just a few days, because we can use 1536 well plate format with a read time of only 7 minutes per plate. So this is highly resource efficient because with 1536 well plate, you only need very little amount of the protein. So we have tested that 200 microgram of the protein is sufficient for us to generate 50,000 data points. So this strategy allows us to have a powerful and scalable way to identify molecular glues with high confidence and minimal resource consumption. So for the spectral shift assay, we are using an instrument from Dianthus platform from Nanotemper. So this is a cutting-edge microfluidic-free solution for affinity screening that leverages on a unique detection method called spectral shift. Okay. So what makes spectral shift special? So it's a dual wavelength detection, specifically at 650 and at 670 nanometer. So within a stable isothermal environment. So binding of a ligand onto the protein will induce a shift in fluorescent intensity of the labeled protein, or V1. So either to the left or to the right of the wavelength, depending on the charges on the surface of the protein. So this ensures highly sensitive and reliable measurement without the complexity of temperature fluctuation or microfluidic systems. So the assay is designed to be simple. We only need one protein to be labeled with a fluorescent dye probe. So this keeps the system close to native condition, which is ideal for preserving biological relevance while still achieving high sensitivity. So in terms of throughput, Dianthus is a powerhouse. It can generate up to 65,000 data points per day, so making it perfect for large-scale screening campaigns. So plus it's compatible with 384 and 1536 well plate format, so giving you the flexibility of depending on your assay needs. And to streamline your workflow even further, our data analysis software is fully automated for high throughput screening. We use the Gene Data Screener platform for data analysis. So this platform can integrate seamlessly with any HTS platform to deliver fast and reproducible data analysis and insights for high throughput screening data. So overall, our in-house setup is designed to maximize your resources while maintaining exceptional screening efficiency and data quality. Okay. So now let me walk you through how we identify molecular glues for V1 using a structured screening cascade. So we started with high throughput screening, miniaturizing it down to 1536 well format to enable efficient and economical screening. From there, we screen a focus degrader library of 106 compounds. So this library was built from a commercial library called MacChem Express that we purchased from molecular glue library. And also we add in all the ProText that we have synthesized for our internal case studies here on PIM3 and V1. Yeah, I will talk about PIM3 later. So in this case study, we use ProText that we have designed and synthesized for both PIM3 and V1 case study. So, so there are more than 100 compounds here, and we also spike in a few known literature compounds that are described as V1 degrader, for example, S36 and 40I4 here. Okay. So once we have the hit here, we will use the same spectral shift assay to confirm binding by doing a dose response curve. Okay, and we determine both binary and ternary complex affinity. We profile these hits also using in-house kinase profiling platforms like KinoScan and also kinase profiler, and to access V1 selectivity and inhibition. And we also have an in-house E3 scan platform to determine whether they are binding to other E3 ligases besides cerebron. Next, we run a tier 1 ADME package to evaluate drug-like properties, following by a cell-based assay to measure V1 degradation via Western blot. So finally, we validate the degradation effect at the proteome level using whole cell proteomics in HepG2 cells. So, okay. So now we look at the actual data, actual results. So we screen a focus library of 106 degrader across 3 different conditions, V1 alone, cerebron alone, and V1 cerebron in combination. So these are shown in the primary screening, the results are shown in the primary screen section, where you can see the response profile for each condition. We screen at a single concentration. So most of the compound, of course, doesn't hit. So it's in the purple line here. So our positive control is away from the purple line here in yellow, and then hits compound are in light blue. So these are the hit compound. As I mentioned just now, our library has a lot of ProText against using CRBN, cerebron as the E3 ligase. So when we are screening against cerebron, we get lots of more hits compared to if we are screening against V1. Yep. So these hits, the light blue color dots are the hits, and they are, we consider them as positive or negative hits. But we consider them as a real hit or positive hit if there are 3 SD or 3 standard deviation away from the compound with no activity. Okay. Next. Next, following the primary screen, we move on to hit confirmation. So here we validate the hits by generating dose response curve, which allow us to calculate binding affinities, KD, and importantly, calculate the cooperativity factors for ternary complex formation. So to visualize the overlap and specificity of the binders, we use a Venn diagram. So you can see that 37 compounds here bind to CRBN or cerebron alone, and 6 compounds binds to V1 alone, and 7 compounds bind to V1 and cerebron complex. Yeah, and not to V1. They just bind to the complex, but not to V1 alone. V1 is bind to complex and okay, I explain it again. So we have 1 compound that binds to both cerebron and V1, and 7 compound that doesn't bind to V1 alone, but bind to the ternary complex for V1 and cerebron. So this is the interesting part. So they are the potential molecular glues. So this overlap here is critical. So it helps us understand the mechanism of action of each of the compound and distinguish between binary binders and those that truly promote ternary complex formation. So overall, this workflow not only confirms hits, but also provide mechanistic insight, making it a powerful approach for molecular glue discovery. So now we take a look at the closer at the data. So when we are screening for either the binders for cerebron or V1, this is what we get, and we plot the KD value for V1 and cerebron on different axes. This is what we get. So we identify compound that binds strongly to cerebron or compound that binds strongly to V1. Yeah. There is only one compound that binds to both cerebron and V1 strongly, and this is a PROTAC based on a double study that I mentioned just now. Yeah. So we didn't identify anything here. And this is from this chart, it looks like we didn't identify any molecular glue or other PROTACs. Yeah. So now, how do we identify molecular glue in this case? So we revisit the same data set, but this time we plot the ternary binding constants on the x-axis and the cooperativity factor on the y-axis. So this shift in perspective reveals a completely different profile. So the V1 cerebron PROTAC is still present in this analysis, but it shows a cooperativity factor of only 0.5 here against V1. Okay, this means that the ternary complex formation for this PROTAC is actually less favorable than the expected based on the binary interaction. The ternary complex of a large KD value compared to the binary KD, right? So but we want cooperativity factors that are higher to show that it actually brings the ternary complex together better. So how do we do that? So we can see that this line here in the middle, they have better cooperativity factor and also better KD value. So the compound here, you can see that BMS compound, this is a molecular glue. It has a cooperativity factor of more than 88, and it doesn't bind to V1 alone, but it has a ternary complex KD, measurable ternary complex KD. But the important part is it doesn't bind V1 alone, but it brings, when CRBN is present, it brings both together into a ternary complex. So these are the molecular glues that we want to identify. So what is more exciting here is we actually discover several molecules that are even better than the BMS molecular glue. So, and we can tell from there that they have no binding to V1 again, but only binding to cerebron, but they bring the complex together, the two proteins together with a better KD values than the known BMS drug. So this is, we have identified some novel molecular glues here. So this opens up a new avenue for exploring non-traditional binders. So in short, successful molecular glue binder discovery isn't just about binding. It's about forming the right ternary complex, in this case, at the right concentration with the right cooperativity factor. So this is what we want to emphasize here. So one of these compound here, we take one of this compound here and we do further studies. First, we confirm its binding to V1 and cerebron using orthogonal assays. So we use our own in-house kinase selectivity platform called KinomeScan. So we shows that our compound has no significant binding to V1 and also to a V1 analog known as V1b. So it's also called V2 actually. So it has no binding to either of these V1 molecule. And then we use spectral shift assay again to access the cerebron binding. And we observe a KD of 15.6 nanomolar. So this indicates a strong binding to the cerebron. So to validate this, we run our proprietary qPCR-based E3 scan platform, which confirms binding to the cerebron and the cerebron DDP1 complex with a KD value of 25 nanomolar and also 8 nanomolar. So the compounds bind to CRBN DD1 a DDB1 complex better than CRBN alone on our E3 scan platform. So this supports that the compound's ability to engage E3 ligase complex. Okay. Then we also perform cellular target engagement to show that the compound can enter into the cells and still bind at the cerebrum. Okay, as expected, the IC50 value here is 10-fold higher than the value here in the binding assay. So this is taking into consideration membrane permeability and accessing to the subcellular compartment where the cerebrum is located. So the graph in the middle shows the complex, ternary complex formation. So binary, there's no binding. Ternary complex formation, we have a binding and with a proper dose response KD binding curve. So this shows that our compound promotes selective interaction between V1 and cerebrum, yeah, and without so promoting ternary complex formation in an acellular system here. So far, we have been doing it outside of the cells. Does not necessarily guarantee a degradation of the protein of interest. Therefore, we conduct further profiling of our molecular glue to assess its functional activity. So on the right side, we use HepG2 cells treated with the molecular glue for 24 hours, and V1 levels was assessed. Yeah. You can see from the Western blot results, the V1 protein ranges from 3 nanomolar to 3 micromolar here, and each concentration was tested in duplicate, and as the dose of the drug increases, we observe a clear reduction in E1 signal, V1 protein level, especially at the very high concentration, yeah, of the compound. So this confirms that our compound is not only active in the biochemical binding assay here, but also function effectively in a cellular context. So promoting V1 degradation in a dose-dependent format. So the volcano board down here, volcano plot down here, represents the proteomics profiling results of our compound conducted in HepG2 cells. The cells were again treated for 24 hours, and analysis were performed using our Orbitrap mass spectrometry with data processed through Protein Discovery의 software. So on average, we identify about 6,636 proteins, giving us a broad view of the cellular response. Okay, so we did not detect V1 in this data set. So this is likely due to low expression of V1 in a HepG2 cell. But this data is still useful, and we continue to analyze this data to confirm any indirect effect. So for example, GSP1, this is a known off-target for many molecular groups. So it's especially molecular groups derived from thalidomide, which bind to cerebrum. So this is important that this is not affected. So we support that the compound is selective and specific. Then tubulin beta-3 chain is a key component of the microtubules. So it's significantly downregulated here, so showing that it's an inhibition of cell mitosis, as we expected. Yeah. So to improve selectivity of the V1 detection of V1 protein in the cells in the future experiment, we are now planning to use cell line like MV4-11, which have higher V1 level compared to HepG2. So we'll repeat this proteomic analysis again, but now it supports that the cell is, the compound is selective. It doesn't non-specifically affect the GSP1 protein level, and it also induce reduction of tubulin levels. So in summary, this is a highly selective V1 molecular group validated across multiple different types of orthogonal assays and platforms. So to wrap up this case study, before we go to the next case study, let's take a look at the key learning points where we are headed. Where we are headed next. So first, this project clearly demonstrates how high throughput spectral shift technology with Eurofins Discovery can bring real innovation to molecular glue discovery. The platform allows us to screen efficiently and identify a diverse set of compounds, including cooperativity and non-cooperative binders, all in a single run targeting the V1 cerebron complex. We can run 3 different experiments in parallel, so it's like a single run where we identify all binary binders and ternary binders. So this affinity-based approach not only streamline hit identification, but also give us direct insight into the binding dynamics and cooperativity factor, which are critical for understanding molecular glue behavior. So looking ahead, we have a few important steps to progress. So we are working to complete the protein identification from the proteomics study to better understand downstream effect, especially using a cell that has more expression of V1. Okay, and then since ternary complex formation doesn't guarantee translation to protein degradation, we will conduct more cellular profiling of the V1 molecular glue candidates to confirm their functional impact. So together, these efforts will help us refine our understanding of the molecular glue mechanism and also move promising candidates closer to therapeutics relevance. Okay, to summarize again, this slide compares 4 different screening strategies for molecular glues and binder discovery. So let's start with the protein requirement. Spectral shift assay is highly efficient. It only uses 1 milligram of protein, which is enough for you to screen 100,000 compound library plus confirmation by dose response. So this is significantly lower than other ASMS or affinity-based mass spec, which require 100 milligram protein or HTRF or DEL protein screening. So in terms of protein targeting, SPS is flexible. It works with both tagged and untagged protein if your protein is autofluorescent. So this is a major advantage over the DEL or DEL approach, which strictly require that the proteins are tagged and immobilized. Whereas our assay, SPS or spectral shift assay, is in solution, so there's no restriction to immobilizing of the protein, which may deform the protein. Yep. So this is a major advantage, and also simpler setup because it's performed free in solution, free floating protein in solution, and preserving the native protein behavior. Okay. So when it comes to compound library, spectral shift is compatible with any library and uses a straightforward 1 compound per well format. Okay. In comparison, ASMS and DEL relies on pooling strategy or DNA tag library for DEL. So this can complicate hit deconvolution and downstream validation. Next, readouts. This is where spectral shift really shines. So it provides a direct binding measurement, allowing us to determine KD values for both the binary and ternary complex simultaneously, as well as calculating the cooperativity factor. For other methods like HTRF and DEL, so they rely on indirect readouts like PCR readout, which often requires additional orthogonal assays to confirm binding. And also, compared to spectral shift assays, ASMS, DEL, and HTRF tends to have more false positive results compared to spectral shift assay because of the less manipulation, and also we only need to label one of the protein. So, and then talking about timeline and outcome, the timeline here referring to assay development to hit confirmation. Spectral shift assay only takes about 2.5 months from assay development to screening to hit confirmation, whereas other approaches will take more than that, sometimes up to 6 months due to the complexity and deconvolutions of target and so on, deconvolution of the hits and so on. So overall, spectral shift assays offer a powerful combination of speed, sensitivity, flexibility, and direct data output, making it an ideal platform for efficient molecular glue discovery. So Eurofins, besides the spectral shift assay, Eurofins Discovery has three powerful biophysical techniques to measure the affinity and stability of ternary complex. They are critical for understanding how degraders function at the molecular level. So starting with SPR here, this method allows us to monitor complex formation in real time, just like SPS, but the protein needs to be immobilized. So this is the shortcoming for SPR here. Yep. Next, we have the micro-scale thermophoresis or MST using trick C of the spectral shift that I mentioned just now. Yeah, so these are solution-based and mass independent. So meaning they work well even with large ligases. One of the protein needs to be labeled. Usually we label the protein of interest. Yeah, and a spectral shift assay is the same as SPR. We can detect Hooke effect at high concentration of the protein. Yeah. Finally, we have the isothermal calorimetry or ITC to characterize the thermodynamics of ternary complex formation. So a negative Gibbs energy combines with a positive enthalpy change indicate a stable complex. So ITC also confirms the 1-to-1-to-1 stoichiometry of the target, degrader, and ligase, essential for validating the mechanism of action. So together, all these techniques provide a comprehensive view of how degraders engage their targets and E3 ligases, helping us to design more potent and selective degraders. Okay, so now we move on to our second case study focusing on PIM3 kinase. Okay, so instead of traditional inhibition, we explore PROTACs to achieve targeted degradation. So the process involves two key steps. First, identifying ligands for PIM3 and suitable E3 ligase binder. Then characterizing the resulting PROTACs for selectivity, potency, and degradation efficiency. So this approach demonstrates how rational design, like linkers, exit vector, and systematic screening can turn challenging kinase targets into a degradable ones, thus opening up new therapeutic possibilities. So when designing a bifunctional degraders, we don't have to think of these steps, the three steps here, as sequential. In fact, all three categories, optimizing MedChem design, virtual screening, and high throughput screening, can run in parallel to accelerate discovery. So on the left, we have the optimized MedChem design. So this involves literature to lead strategy, leveraging experienced medicinal chemistry approach, and even applying AI for design and prioritization. So at the same time, we can perform virtual screening. So here, a computational chemistry tool allows us to screen a massive virtual library of 27 compounds and using AI for protein structure and heat selection. And in parallel, we can execute affinity-based screening or the traditional high throughput screening. So we have the ability in-house to screen over 700,000 compounds with different kinds of library, diversity library or marketed drug approved library or GPCR library, or even a fragment-based library. And we can also perform DEL or DNA encoded library to our partner company. So by running all these three streams together, we maximize efficiency and increase the chance of identifying novel modulators and binders quickly. So this integrated approach here is key to accelerating bifunctional degrader design. How do we do that? So this slide illustrates our approach to design and profiling PROTAC array targeting PIM3 kinases. So we start by selecting a diverse set of PIM3 inhibitors that we identify through our high throughput screening platform and from literature. So these are then paired with various linkers differing in length and chemical structure, chemical nature, and combine them with E3 ligase binder for cerebron and VHL. So the goal here is to generate a broad array of PROTACs with different structural configurations to explore how this variation affects the degradation efficiency. So to streamline this process, we use virtual library enumeration and AI modeling to prioritize compound. So the graph here shows how we use AI to narrow down candidates based on their predicted ADME properties, so leading to focused selection of compound for synthesis. So this can be done in-house with our proprietary Sapphire suite of AI models. So the strategies here allow us to rapidly explore SAR or structural activity relationship studies and identify the most promising degrader for further development. So this slide shows how we use computational modeling to guide the design of the bifunctional degraders. So specifically, we want to identify an optimal exit vector on the protein of interest. So on the left here, you see the crystal structure of PIM1 kinase in complex with the AZD compound called AZD1208. There's no PIM3 crystal structure, so we use PIM1, which shows high homology to PIM3 as the model structure. So we analyze how the molecule binds and where we can attach a linker, the exit vector, without disrupting its interaction with the target. The arrow here shows the exit vector, the position on the molecule where we can extend a linker towards the E3 ligase binder. This is a critical design decision as it affects the geometry and stability of the resulting ternary complex. On the right, in this example, we have applied this insight to design a PROTAC targeting PIM3 using cerebron as the E3 ligase. The resulting complex, we call it 154-1 here, incorporates optimized exit vector and a linker length to ensure productive ternary complex binding. So this approach helps us to rationally design degraders with improved efficacy and selectivity, and also reduce trial and error and accelerate lead optimization. Within Eurofins Discovery, we have the ability to synthesize chemical compound quickly. So this slide outlines the chemical synthesis of one of the compound for our PIM3 targeting PROTAC. So the synthesis starts with a commercially available bromoaldehyde derivative and proceeds through the 5 steps here using standard organic chemistry techniques. We first build the AZD1208 warhead here, which target PIM3 and then connects it to a CRBN binder. In this case, the CRBN binder is a pomalidomide, okay, via a tailored linker. So this linker has been designed and tested to be good for the binding of the ternary complex. So the assay conditions and reagents are shown here, including the palladium catalyzed coupling here. And then base-mediated transformation, and finally purification by preparative HPLC. So this streamlined route allows us for efficient production and optimization of the PROTAC compound, so enabling rapid iteration during the lead development phase. In Eurofins Discovery, we leverage automation to accelerate PROTAC synthesis. Using our robotic lab, we synthesize an array of over 70 PROTACs, combining different PIM3 inhibitors, linkers, and the E3 ligase binder, specifically cerebron and VHL. This automated approach allows us to rapidly explore multiple exit vectors on the PIM3 kinase site, while simultaneously optimizing the linker length. So linker length optimization is important because it's a key factor in the ternary complex formation and the degradation efficiency. The integration of the robotic synthesis not only increase throughput, but also ensure consistency and precision across the entire compound set, helping us to move quickly from design to profiling. So this is our well-established workflow for compound library synthesis, built over 20 years of experience. The process begins with solid or liquid dispensing, followed by careful controlled reaction condition. After that, we move on to extraction, evaporation, purification, and ending with lyophilization, and finally compound handling. So this streamlined workflow allows us to deliver 7 to 20 milligrams per target per compound. 7 to 20 milligram compound. This is enough for full quality control, physicochemical analysis of the compound like solubility, log D, tier 1 ADME profiling, and in vitro screening. So with this setup, we can synthesize and process up to 50 compounds per week, making it ideal for rapid SAR exploration and lead optimization. So I'm showcasing a selection of PIM3 targeting proteins that we have successfully synthesized as part of our design and profiling campaign. So each compound here labeled these numbers here represent a unique combination of 3 core components. A PIM3 inhibitor in green, a linker in the middle, and a E3 ligase binder in red on the left-hand side. Okay. You can see this compound color-coded help us to visualize how different configuration influence degradation potential. So this modular design approach allows us to systematically explore in parallel variations in linker length, exit vector position, and E3 ligase selection. Yep. So with the ability to quickly synthesize and profiling this compound, we are able to do rapid SAR studies to optimize and identify which combination gives the most effective ternary complex formation and target degradation. So again, we always compound a screening cascade for our client to identify the most promising proteins. So and this cascade will help us to move quickly from synthesis to biological validation. So we begin with MedChem synthesis, MedChem design and automated high throughput synthesis. This is followed by biochemical assays like PIM3 inhibition using ATP glow and cerebron binding via E3 scan. Next, we also run a tier 1 ADME assay, assessing the solubility, log D, permeability, and metabolic stability in both rodent and human system. In the yellow part here, we dive into the biophysical characterization using spectral shift to evaluate binary and ternary complex formation. So in the green part, once we have promising binders, we access cellular target engagement with nano-bread system or monitor PIM3 degradation in cells by Western blot. We can also run selectivity assays to check for off-target effect in purple here, and rodent ortholog activity and confirm protein degradation by Western blot again. Yeah. Further down, we can, for advanced profiling, we move on to tier 2 ADME. So this includes cytochrome inhibition and induction, cardiotox liability study, HERG study, cellular phenotypic assays to measure downstream biomarkers associated with protein degradation. So finally, in the yellow part, we conduct rodent PK/PD study measuring protein levels in specific tissues for oral or IV dosing. So thanks to this streamlined workflow and turnaround time of less than 10 days per assay for most of these assays, we are able to rapidly identify and prioritize the most promising proteins for further development. On the right side, we are able to test and rank the efficacy and preclinical properties of many compounds because we can do automated parallel synthesis, as mentioned in the previous slide, to generate many different proteins to fit into this screening cascade here. So let me do a brief detour of the case study for a moment. So this slide showcases the breadth and depth of Eurofins Discovery's off-the-shelf assays. We offer more than 10,000 ready-to-run assays, spanning binding, biochemical, and cell-based assay. Okay, so this assay cover a wide range of target classes, as you can see here, and are available for a very rapid turnaround time, typically just 10 days or 15 business days per assay. So I would like to bring your attention to the kinase and the enzyme part, since we are working with a PIM3 kinase in this case study. So we offer more than 2,500 assays for kinase and enzyme in combination. This cover both binding assay, functional assay, cell-based assay, and also cell-based target engagement assay. So all these assays are ideal for early screening, selectivity, and mechanistic study. So for kinase study, we offer all this stuff. And then And all these techniques can be measured with a variety of measurement technology like radiometric binding assay, mass spectrometry, luminescent fluorescence, PCR-based biophysics, and even high content analysis. So all these assays here are backed by a team of dedicated experts who provide scientific guidance and support throughout your project. So whether you are screening, profiling, or validating hits, our assay portfolio is designed to help you move quickly and confidently from idea to drugs. So coming back to the PIM3 case study, so and making use of our extensive portfolio of off-the-shelf assay on the previous slide, we perform biochemical and cellular characterization of the binary complex formed from between PIM3 kinase and also with CRBN E3 ligase induced by our PROTAC candidates. Okay, so starting from kinase binding data on the left, we use both kinome scan, which is for binding, measuring binding, so we give you a KD value, and also for kinase profiler, which measure activity of the kinase. So it gives you an IC50 of inhibition of the function of the kinase. So 3 of our PROTAC candidates here, and we measure it against 3 different kinases, 3 different PIM kinases. So you can see variation of PIM3 cross-reactivity here for our compound. So on the E3 ligase side, we access binding to cerebron and cerebron DDB1 complex again using our in-house E3 scan platform. So these measurements are critical for confirming that our compounds can selectively recruit E3 ligases. So you can see that generally binding to CRBN alone is slightly less strong compared to binding to CRBN DDB1 complex. Then coming back to cell-based assay, the compound also show cell-based assay, sorry, using ATP glow assay for functional assay to validate our kinase profiler assay. We show that our compounds show 23 to 38 nanomolar binding, indicates strong biochemical inhibition of the kinase function. Then moving on to cellular assays, we compare it with intact cell and with permeabilized cell. So not surprisingly, in intact cell, the PROTAC, which is big, is harder to enter the cell. So you have an increase in the 10-fold, usually about 10-fold or more increase in or decrease in the IC50 compared to in the cell-free environment. However, in permeabilized cells, you can see that the IC50 has improved a lot. So this shows that the compound doesn't generate, the PROTAC compound doesn't enter cells so easily. So we can have all these assays to measure this and to confirm this effect. So all these data collectively help us to prioritize compound with strong binding, good cellular activity, and also favorable permeability profile for further development. So here we perform spectral shift analysis of the compound, of one of the compound, focusing on its ability to form binary and ternary complex with CRBN and PIM3. So on the left, you see the graph showing the fluorescence ratio of 670 over 650 for the spectral shift. So from the data, we calculate the KD, and the KD is 31 micromolar nanomolar for the binary complex and 45 nanomolar for the ternary complex. This gives us a cooperativity factor of 0.7, which suggests actually a negative cooperativity, which means the presence of the PIM3 is slightly weaker than the overall binding. So the low cooperativity indicates rapid dissociation kinetics and suboptimal interaction between PIM3 and cerebron when breached by our compound. In other words, the compound binds well individually, with 31 nanomolar and individually to the PIM3. But when it comes to ternary complex, it actually weakens the interaction. So this is crucial to guide further optimization. So whether by modifying the linker or adjusting the exit linker or exploring alternative E3 ligase, so will help us to improve this. I will show you in the next slide. So this is our other improved PIM3 bifunctional degraders. So we're characterizing them using biophysical and cellular assay. So for biophysical assay, we use spectral shift to access the ternary complex formation. So on the top graph, you see two curves, one for the binary interaction with cerebron and the other one for the ternary complex formed between the green one formed between all three, cerebron, PIM3, and the compound. So the dissociation constant here tells a different story, right? So the binary complex to PIM3 is 81 nanomolar, while the ternary complex has reduced to 31 nanomolar. This gives us a cooperativity factor of 2.68, suggesting a positive cooperativity in the presence of the ternary complex. So another compound here. Again, we see that the binary binding gives you a 100 nanomolar KD, but the ternary complex formation gives you a 34 nanomolar KD. So this shows a cooperativity factor of 2.99. So again, this shows that this compound, our two compound here, is an improvement over the compound in the previous page. And we can also show there is a hook effect here for this compound. There's no hook effect for this particular compound using a spectral shift assay. So hook effect is because when there's a constant increase in concentration of the PROTAC beyond an optimal point, it actually reduces degradation efficiency. Right? As we can show here in Western blot, there is a degradation of the compound at a low concentration of the PROTAC, but as the concentration of the PROTAC increases, the degradation of the protein actually decreases. So this is because the binary complex, the PROTAC molecule at high concentration start to form binary complex. So binding either to the target or to the E3 ligase rather than producing the ternary complex. Right? So this data again confirm that PROTAC, we can effectively design PROTAC that form competent ternary complex and induce PIM3 degradation. So the key learning point here, the robotic parallel synthesis. First, we use robotic parallel synthesis combined with efficient screening cascade to significantly accelerate the identification of promising PIM kinase degraders. This workflow has proven to be both scalable and effective. Second, we have learned that understanding the principles of the ternary complex recognition is absolutely critical. It's not just about binding affinity, it's about how the complex come together in a productive complex. This insight is now guiding us in our structure-based design of PROTAC. Third, we have shown that the hook effect, which is often a challenge in degrader drug development, can be observed using spectral shift biophysical assay. So this gives us a powerful tool to anticipate and mitigate this issue early in the design process. Looking ahead, we are focusing on two main areas. First, we will complete the full profiling of our most promising compound to validate their efficacy and selectivity. Second, we will leverage the insight from this PIM3 case study to inform the rational design for PIM3 molecular glue. So this case study is for PROTAC, and now we have another ambition. We want to design PIM3 molecular glues. So these are exciting directions that open up new therapeutic possibility. Okay, I'm coming to the last two slides. So this slide brings everything together, summarizing everything that Eurofins Discovery's comprehensive support across each step of the targeted protein degradation workflow for both molecular glue and PROTACs. At the top row, you can see bifunctional molecule design, where our medicinal chemistry, computational chemistry, and synthetic chemistry teams alongside with robotics and high potency lab help design and optimize degraders. Next, we move on to target and E3 ligase binding and ternary complex formation. So here we use advanced biophysical tools like spectral shift assay, SPR, or MST to screen and characterize molecular interactions and supported by qPCR platform like E3 scan. Next, we access cellular target engagement and ubiquitination using our in-house, which I didn't show in these two examples, using our in-house assay known as in-cell hunter and a bread-based assay to understand mechanistic insight into the physiological context. For target degradation, we also can rely on our in-house binder technology, a JAS Western blot, JAS capillary and automated Western blot instrument, and mass spec-based proteomics to identify or quantify protein turnover and selectivity. And finally, we evaluate cellular function through our large portfolio of phenotypics assay, ADME, and safety screen panels that I mentioned earlier to understand downstream effects and safety profiles. So together, these solutions form a robust end-to-end platform to accelerate and de-risk your TPD programs. So this is the typical workflow that we put together after discussion with the client. We offer medicinal chemistry and synthetic chemistry design. And the second row here, you can see that this is after we have the compound, we follow by cellular target engagement and degradation assays. We also recommend biophysical assays in the fourth row here for the ternary complex formation to understand the binding kinetics. Yeah. This binding kinetics will also inform the chemist to optimize the degrader compound. Then on the fifth row, in the light blue column, you can see for target specificity and potential cross-reactivity identification, we have in-house phenotypic assays and also biomap and oncopanel to determine downstream biomarkers and genes that are modulated by the degraders. This is followed by our tier 2 ADME assays and also animal PK/PD studies. So we will have a complete picture of the degrader efficacy, selectivity, and mechanistic understanding of how it behaves in our body to help you to select the best candidate to go into clinical trial. So this is another view of how Eurofins Discovery can support targeted protein degradation through our integrated discovery platform known as Discovery One. So you know that Eurofins, we have capabilities in chemistry, ADME, pharmacology, biophysics, safety, tox panel, and in vivo studies and translational models. So on top of this, we have a Discovery One. So Discovery One is a team of experienced drug discovery scientists. They provide program management with deep scientific expertise and guidance to help our clients to deliver high quality, actionable results. So with Discovery One, you get a streamlined, scientific-driven path from discovery to preclinical candidates. Okay, that's the end of my talk. Thank you so much for your attention and interest in this topic. I will be happy to take any questions that you have, and I will do my best to answer them. Thank you.